Good morning, everyone. Today is now 2:08 in the Kingdom of Thailand. So we have the OBG morning activity today, and this is the first time that we are going to host someone from CNMI, and also about the work that she and her team has done will be something that I know very little about. That is why we need to know her and her work for this time. And normally the Wednesday program will focus on topics that are related to or support research mission of our department. I am Dr. Chutima, topipat, your host. Today we welcome Dr. Pimonrat Ketsawatsomkron นะคะอาจารย์she worked on Obgyn-Rama Medical Institute, Faculty of Medicine, Ramathipati Hospital, and her education and training in pharmacy school, Mahidol University, and after that she studied biomedical science, Medical College of Georgia, Augusta, USA. And the research area she interests is about cardiovascular lab, focusing on blood vessel function using multidisciplinary approaches from 2D and 3D in vitro model to whole animal study. And right now she developing blood vessel on chip model for drug and compound testing. So when I look at her publication, there are lots of things, substance, and also factors that we might heard about in our basic science related to the clinic, and we want her to exploit a bit for us what work that she do. And also with her we have another supporting speaker, Ajahn Kenjiro Muta. He also working on Obgyn-Rama Medical Institute, Faculty of Medicine, Ramathipati Hospital, and he studied but applied biochemistry, Saga University, Japan, and after that he studied molecular cellular biology, molecular and cellular biology, University of Iota Carver College of Medicine, IA, and he will be with us online to supporting his wife here. So let's start, and I would like to pass a microphone to Ajahn Pimonrat. Could you please provide us the knowledge and information that will enlighten us that something we can collaborate in the future, please? Ajahn Pimonrat, เชิญค่ะ
Thank you very much, and good morning, everyone. Thank you for having me today, and thank you for the nice introduction. Actually, we can speak Thai, or I will present in English, and if it's a question in Thai or something, don't be shy, just go ahead and ask. Because this is pretty new model, and I think we are the first group. I think we are the only group in Thailand that work on this at the moment, because this technology is only like 10 years old, and it's been increasingly interesting. And I will tell you the story about it and how I got started. Okay, so I'm in a preclinical group, and we are at the CNMI, and I've been here since 2018. I'm very happy to talk to you today, and please do not hesitate to contact me if you ever have any questions or would like to discuss further. Like Ajahn Chutima already introduced about myself, and this is my education background. But I'd like to get into the work experience a little bit. I was in the US pretty long, like 15 years. After finishing my PhD, I actually pursued my postdoc at University of Iowa, Department of Pharmacology. There, I was working in a pretty big lab in cardiovascular research field, where they have about 20 people, range from bioinformaticians up to geneticists and physiologists. Was working there as a postdoc. It was a pretty productive years, and then got promoted to be a research faculty there. I was there for like 3 years. After that, and moved to Seattle. Never thought about going to Seattle, but end up there. Raining the whole time, about 9 months a year. And I was working as a project lead doing blood-brain barrier on chip. So I was there basically got exposed to a new technology, organ on a chip. Nortis was a small biotech company that spun off from University of Washington. So it was pretty exciting to work in a new technology and be able to get exposed to commercialization as well as engineering team. So I really enjoy that. So after working there for a couple of years, I was still in Seattle at that time, but I got a position as a consultant for Siam Bioscience, but I was working remotely for like 7 months. And then again, it was the first time to work for a biotech company in Thailand. It was pretty interesting. I was looking at oversee their non-clinical project where they have their drug testing in another country. So it was pretty interesting. So after that, decided to move to Thailand with the whole family, my husband and our daughter. And we moved here 2018, and I actually sit in Payathai for a year because at that time CNMI wasn't finished yet with the building and all that. I'm sure probably most of you have been to CNMI. This is how it looks right now. This picture probably not updated. We have more building than this picture now. Yeah. I'm sure you guys have heard from my colleague from preclinic group actually came to give a talk here. CNMI have pretty unique positioning because we have hospital right here, and then across the street, our preclinical building is actually here. Actually, there's anatomy building popping up here too. It's not included in his picture. We have pretty close interaction between preclinical scientists and physician. And we also, you know, at the, I don't know, you guys have heard, 7th floor in the preclinical buildings, there will be a upcoming stem cell and cellular therapy center. If you guys ever interested, I'm part of this team, helping them setting up the lab. Ajahn Chakri Gitayagon is the head of this project, and we are basically building up a GMP facility, and hopefully it will be certified and will be one of the first in Thailand that have this facility, having this cellular therapy coming out. In preclinical buildings, in our preclinical group, we are located on the 4th floor and 5th floor. We are pretty well equipped with the molecular and cellular biology, and we have very good people. Ajahn Titiwat and the team actually pretty good at, they are very good and well trained in advanced microscopy. So we build pretty good advanced microscopy capability. We also have some pharmacologists who's interested in mass spec and analytical type of assay. So we have quite a few mass spec there. And we have Ajahn Pisut, Ajahn Bia, right? Who's interested in bioinformatics and system biology. He's very, very productive. So today I will be talking a lot with the drug development process and drug discovery, because initially the organ on chip was built and was established because to solve the problem of the drug development, how inefficient it is, and they are trying to make things better. And it's been changed since the past year because of the COVID, but this is how drug development process look like. Traditionally, it would take about 15 years to develop a new drugs from research perspective until it go to the market. So usually when you want to have a new drugs, usually it start from non-clinical phases, which involve with the basic research, drug discovery, testing in vitro and in vivo, both efficacy and toxicity testing. Then once you have a good candidate, it will be moved to the clinical phases where probably you guys familiar with phase 1, phase 2, and phase 3. And if everything looks good, if you submit to the FDA, if it's Thailand, Thai FDA, US FDA, or in Europe, it will be EMA. once it's approved, then you can sell it in the market. Usually it costs about one drug, it will cost about 2.5 billion US dollars on average. That's why we don't see this a lot in Thailand or in some countries. Mostly it's being done in Western country or in the US or some maybe in Japan. It's very long time. It takes so long, 15 years, very long time. But COVID changed everything. We can discuss about that later. But it's not a very successful business because the failure rate though, even though they spend 15 years, spend $2.5 billion, the failure rate is actually about almost close to 100%. So a lot of drugs actually fail in clinical trials. And it's just not very good process. So the question, how bad drugs fail in clinical trials and why, right? So it depends on the field that you're in. If it's in cancer drugs, it can be up to 97% drugs actually fail in clinical trial. Alzheimer's disease, if you read the news, 99% fail. So it works so well in animal, how come it's fail in clinical, in patients? And unfortunately, like Alzheimer's disease, usually the drug fail in phase 3, where they spend a lot of money and time on it. It's very inefficient. So after this kind of problem happens, people start looking at the reason why it's failed, right? So, and they found that about 60% of the drug fail in clinical trials because of toxicity or not efficacious enough, so insufficient efficacy. And if they look back and actually look into analyzing more data, they found that this kind of stuff could have been caught in the preclinical phases or non-clinical phases. But because of the model that we use in non-clinical phases, like cell culture or animal models, they're not very predictive of what will happen in humans. So there's a lot of worldwide efforts trying to improve the non-clinical models. Okay, so probably you have seen what have been when they try to establish some experiment or something in non-clinical. They typically, right now, they use a regular cell culture, or I will call it 2D cell culture, because basically they grow the cell on a plastic. You probably see in the lab a lot. These have been used for 100 years. It's easy, it's cheap, it's fast, right? But it's not very predictive, right? Because our cell is not laying flat like this, right? Our cell is interacting with other cell type, and also it has a 3-dimensional structure, right? In vivo, though, if you see, from fruit flies to zebrafish to rodents to sheep and dog and primates, right? And more drugs, new drugs actually involve antibody, and rodent is basically useless. It doesn't predict anything antibody like in human. So a lot of people have been talking about this, and they're trying to improve this non-clinical testing. So I just want to recap what I just talked about. In the 2D conventional cell culture, it lacks physiological context. It doesn't have tissue architecture. It means it doesn't have like 3-dimensional structure, not like in human or in vivo. Importantly, this cell in 2D, it never exposed to shear stress or the luminal flow, right? Where in our body, we have a lot of fluid and a lot of exposure to the flow, right? And also the interaction between cell to cell contact or cell to extracellular matrix contact. So in 2D cell culture, you never have this. So there's a lot of studies showing that the cell in 2D have completely different gene expression profiling compared to the 3D cell. So these people have already shown that in the 2D cell culture, it failed to adequately resemble or mimicking respond to the drugs compared to in vivo. But people do animal model a lot, and I wouldn't say animal model is not important. It's still important, but it just have its own limitation, and it's so time consuming and high cost. And it's species differences. Mouse, rat are not human. So basically it cannot adequately represent human diseases. And also ethical concern. It's becoming more and more obvious that people don't want to do animal study. So particularly in Thailand, a lot of students that I have been talking to, nobody want to do animal work because they feel bad, right? So there must be a new way to improve the drug development process, right? This is just an example, the table here showing toxic concentration in micromolar, in vivo, in vitro, 2D and 3D. This is hepatocyte, it's a liver cell, and they're looking for toxic concentrations. And here is the different drugs, and this is plasma concentration in rat that cause toxicity. And you can see here that giving an example like chloroquine, 2.5 micromolar will cause in vivo will cause the toxicity in the liver. In 2D, they tested it and it's about 5. When they do the 3D, I'm sorry, it is close to, it's similar to in vivo. This may not seem obvious, but if you look at this drug, for example, isoniazid, the plasma concentration is about 30 in vivo that's cause toxicity. 2D, it went to 1,100 micromolar, which if you give this dose to this concentration to in vivo, probably the liver probably already toxic. Yeah, in 3D is more predictive of what happened in vivo. So highlighting the significance of the 3D models over 2D. And very recently, I think it's December 20 something, finally, the US Congress actually already passed the new law. It's called FDA Modernization Act. And this changed the whole ball game of the drug development because previously the FDA required that all the pharmaceutical company, when they try to market the new drugs, they need to submit the data generated in animal testing. But now, they use that for like 80 years or so. But now things change because of the problem that I told you about. So the new law actually does not require the animal testing anymore. They encourage people. It doesn't mean they stop the pharmaceutical company. They didn't say you don't do animal testing. You still can, but it's not a mandate. It's not obligated anymore. And they encourage people to use alternative safety testing methods, particularly with a new in vitro model that can be more predictive of what happened in human. And I'm sure that you talk to Ajahn Pisut, Ajahn Bia, basically the computational is actually also counts as a new model to improve the non-clinical testing. Also, some people doing cell reporter assay where they have an fluorescent dye or they overexpress some of the dye that you can use it as a sensor to see where the pathway is being activated in the cell. But today I will be talking to you mostly about 3D cell culture models, which the advances have been made in these past 10 years. And these might seem so new to us, but I think it's becoming more important. Also, the tissue bioprinting, there are some people working on this, but this is pretty, I think it's just the starting point and it's becoming more and more. But the more advanced one will be 3D cell culture models at the moment. So there are different types of 3D in vitro models, but the one that people talking the most about is organoid. There are some people in Thailand that actually work on organoids. I don't know, Ajahn Doss has talked about it. He worked on enteroid, right? Another one is an organ on a chip. Both of them are 3-dimensional structure model. So let me talk to you a little bit about organoids. Basically, organoids is a pretty attractive model because it's a cell renewal stem cells. You can isolate from patients and then grow in a extracellular matrix. And it becomes self-organized. Basically, it can form some 3-dimensional structure. It's basically creating the tissue-like structure and function. And it's become a pretty valuable tool to study cellular mechanism of disease because you can actually isolate these cells from patients. However, the limitations arise because it's a self-renewal, self-organized stem cell. You actually, it's like a globular structure. So there's something inside, it's a 3D, so, but there's no vascular flow in there. And you cannot actually flow immune cell in there. So if you want to look at immune response, it's very difficult. And you cannot measure the nutrient transport or absorption because what get inside there, you cannot get it out. And it's so inconsistent because the globular structure may not be the same size. And the cell, you cannot control what cell is actually being derived from the stem cell that you put in there because, and also the location of the cell. So this consistency become a problem, but stems, the organoid become a valuable source of cell because you can actually, I believe that you can actually just can subculture them and use them and store them. And I think people have advances this technique that you can get all kind of organ in the body and grow the organoid at them.
Can I see that the organoid is look like right now? It look like something built for the prosthesis rather than just only the function.
Right. So that right now, a lot of people trying to get organoid into the chip even because trying to overcome some of the limitation that they have. Okay, so today I will be focusing on an organ on chip because that's what I'm working on. So the organ on chip, the reason they call it is they call chip because the technique that people use in molding this chip is the same technique that they use for computer chip. So they call it a chip. But if you go read the publication nowadays, some people don't use the word organ on chip because it's maybe sound confusing. Some people use the word microphysiology system or MPS, so it's easier. Some people call it biochip. It means the same thing. So, okay. So when I say organ on a chip, I don't mean I'm building the whole organ on the chip. But basically, we are trying to replicate some of the key physiological function of human organ in the chip. So organ on the chip is an engineered 3D tissue that the key word is it has a flow in exposing to the cell, which doesn't happen in organoids. So basically the engineer actually build the dynamic flow and expose the cell that grow in there to the lamina, shear stress. So the cell mimicking some of the function similar to what happened in vivo. And usually we put human cell in there because we want to replicate the key function in human and created tissue microenvironment that is similar to human. We have a very high control of what cell type we put in and what location we put in. So we have control over this, which is, again, different from organoid. And we can, and in our lab we do too, we incorporate the immune cell in there. So we can look at the inflammatory responses. And some people even use this to study like CAR T, look at transmigration and see how the T cell actually go into the tumor. So this become a very valuable tools. And people start doing something very complex, like co-culture with other type of cell, like in the liver, there are multiple type of cell, they put it in there. So the liver cell become very similar to phenotype is very similar to human. The limitation of organ on chip is that it cannot recapitulate the entire human tissue, only some of the key function. We asked and have a very specific hypothesis, and then we tested that hypothesis in this chip. The reproducibility remain a problem for the chip. I have to say this because 5 years ago when I worked in Seattle, the biotech company in Seattle, that chip is really great, but reproducibility is low. That's become a problem because you cannot have an assay that can service the customer, or you cannot sell.
Question about the reproducibility. Well, actually the chip, they should build up in the same model, but do you mean between the chip interchip assay is not come out the same result.
So basically, there's difficulties both in engineering perspective where the chip is not the same every time they build. Sometimes it's happened because as the chip become more complex, if you add more complexity in there, the chip is hard to use and there's engineering problem. So the trend right now, they try to make it easier for friendly user like biologists like us. And don't forget that when you get into cell culture, there's a biological complication as well. So having more complex model doesn't mean more reproducibility, actually is less. So you need to find that balance. And that's why somebody like me come in, because we basically build this three-dimensional tissue. And since I got into Thailand, we very carefully select the design that people have made it. And there's so many design in this world. Actually, the multiple companies provide the chip, but I have consulted with my colleague in Seattle, and we carefully choose the chip that has the most reproducibility that we can get.
Right.
So when we get here, so when I bought the chip from another countries and build the models, we are confident that the engineering perspective is not a problem. So we've been working on it for 3 years now, and I have to say that the reproducibility that we have currently in the lab is pretty good.
So let's say right now in Jacqueline Narupadin, you have design, but the company that who built the chip will be on board.
No,
somewhere.
basically I import those chip, empty chip, and then
do you mean the chip is already made for general, and
yes,
after that
right.
you
It
try
depends
to,
on us to build the tissue in there.
right?
Yeah, so
right.
we have to find the troubleshooting and protocol and all that, and finally we got the model that is workable.
Right,
so,
right. Thank you.
Yeah, and I feel it's much easier to use than 5 years ago now, the chip, but it's hard to change people from 2D to 3D. 2D is so easy. Yeah. So chip is still difficult to use for general user. And throughput, you cannot do like 384 well plate like in 2D. So it's different. It's cannot be that high throughput yet, but I don't think it needs to be, but I think eventually they will try to push toward high throughput, but it's still difficult. Yeah. Oh, I just want to show you here what are an organ on chips out there. I have to say that in past 5 years, the organ on chip field is really taking off. There are many more models than what I'm listed here. I just want to tell you about the key model that the lung on a chip is the first organ on chip that's been published by a group in Harvard by Wyss Institute. Basically, they grow this epithelial cell on the this chip that they design, and they actually connect this to the vacuum to mimic the breathing rhythm. And they found that this epithelial cell from the human lungs actually mimicking and secreting some of the mucus and all that, similar to what happened in human lung. They got very nice publication out, and people were like, wow, this is happening, right? So then later on, they built a different model. They also have a biotech company spun off from Harvard called Emulate. And Emulate got a lot of money, and they're a forefront in the whole world doing this organ on chip. This is a company I worked with before. They are very good at kidney on a chip. They collaborate with the University of Washington, getting the proximal tubule from patients and put it in here. And they have a series of publications that's very interesting. Then the guy who built the lung on chip when he was a postdoc at Harvard moved to UPenn, and he's actually a Korean guy. Very, very smart guy. He built a lot of interesting model like this one, eye on a chip, because eye is so hard to model, right? In vivo, it's very difficult to test the drugs on it. So he built this eye on chip, And he found that the blinking actually is very critical for eyes development. And actually, I think they are currently using this model for the ophthalmology, for the drug testing, the eye drug testing. So I thought it is very helpful. Brain on chip, people have done it. Heart on chip as well. Let's see, I can get this video to play. Pointer option. No, okay. How do I go back to, or I have to unclick this? Okay. So you can see, I don't know, you can see, look like my cardiac myocyte is actually bleeding in the chip like that. Yeah. So this is big for pharma because they need to screen that drug that has no toxicity to the heart when they're screening for drugs. And the one that people in cancer field really want to do is building this tumor microenvironment on the chip because it's hard to do in vivo. Yeah. So the current application of the organ on chip that is being used in pharmaceutical right now is people try to use it for organ toxicity. It's easy to look at the barrier function in the chip. I have some data to show you today a little bit. And also people look at the transporter, it like proximal tubule a lot on the chip. Because a lot of important transporter is actually being lost after you culture in 2D for a while. Yeah. And like I said before, immune cell tissue infiltration can be easily assessed in the chip. Also, a lot of people in the lab right now is not really being used in pharma that much yet. Is trying to develop the pharmacokinetics, pharmacodynamics across tissue where they connect different organ on the chip. And I have some picture later, and they're trying to see how the drug is being metabolized and all that. Eventually, they're fantasizing that they want to use it for like clinical trial on chip. I don't know when that's going to happen, but that's what they are trying to do. In summary, the advantage of organ on chip in drug testing is that hopefully they can reduce time and cost of drug development and increase the efficiency. Avoid some of the side effects that might happen in human. And also they're talking about using it for personalized medicines where you can grow some of the patient cell into this chip, and maybe you can test the drug before the drug get delivered to the patients. This just want to give you an overview that what happened in pharma is that previously our drug targets is mostly small molecules. But nowadays the portfolio have changed. Mostly the drug nowadays is a cellular therapy or even targeted therapy. And the assay that we have to use to test those complicated targeted therapy drugs or cellular therapy is not going to be something simple anymore. It needs to be something that have capacity to mimic what happened in human better, and start talking about immune response and all that. So we really need this three-dimensional structure tissue to test those drugs. This may not be relevant to you, but basically they are proposing that the organ on chip can be used from the beginning until the clinical trials at the different stages. I do not believe that personally, and I think a lot of people do agree with me that we may not be able to replace animals with the organ on chip entirely, but we will slowly reduce and refine the use of the animals. What else can we do with the organ on chip besides drug testing? Even though organ on chip was built to address the drug development processes, you may not believe, but this is happening, and it's been happening for 2 years now. They use organ on chip to simulate some of the disease condition. NCATs, part of NIH, actually work with DARPA. They bring this organ on chip, different organ, to the space station. This is the real picture where the astronaut is holding the chip from my old company. They were doing kidney on the chip on the space, because they know that some of the aging like disease happens a lot when astronaut came back from the space because of the impact of the zero gravity or microgravity can induce some of those, accelerate those aging process. So if you understand the aging process better, you may be able to understand the diseases actually happening on Earth. But doing the aging study on Earth is pretty long and expensive. So they're trying to accelerate some of the aging process in the spacecraft. So firstly, they use this organ-on-chip, and it's interesting that they increasing the number that go into the space right now with the organ-on-chip. So multiple universities have the model actually submitted. Yeah, so surely the organ-on-chip was being used to test the viral infection in terms of COVID-19 and Ebola. When you want something very quick, organ-on-chip can do that. Also, some of the unethical experiment like exposing radiation or some of the bioweapons or toxin, and look at the impact of this toxin into human body. Of course, you cannot test that in human, and so people start using this organ-on-chip to test some of those toxins. This picture is showing you the impact of the radiation on human gut-on-chip. You can see it's pretty clear to our eyes that radiation is making this gut abnormal, right? So, like I talked to you earlier, they are trying to build a body on the chips. This was published in 2020, and I think it's still majority is still in the lab, not in pharmaceutical scale or manufacturer scale yet. But you can see how things work here. They connect all kind of organ here, trying to see when they administer the drug to patients, where the drugs actually goes and what's come out, and looking at the impact of, metabolism and all that too, trying to establish PK/PD with the body on the chips. So the engineering is fantasizing this, and I think it's pretty interesting. Talking about personalized medicine, they're hoping that this organ-on-chip can be used, for example, testing the drugs in prenatal testing, prenatal group. It's very difficult to test the drug in pregnant women, but if you test that in the chip where we can get the cell, or maybe next slide, I will show you that the model that being used right now with the fetal maternal side and looking at the drug crossing placenta and all that, it can be done in a chip. Also in a child population or children or kids population, we always wonder how the drugs actually respond in those kids. We can actually test that in a chip as well. Or get into very personalized, some group or genetic disease that maybe it's hard to get those kind of cohort. Maybe you can do it in the chip as well. So I looked up last night what other organ-on-chip that could be very useful for OBGYN. Basically, 2017, this group in University of Illinois, Chicago, actually built this reproductive tract 28-day menstruation cycle. Pretty interesting, and they call it IVATA model. If you're interested to look at it, it's pretty complicated model, but there is in a NIH website as well that they are advertising that, oh, how great it is. You can actually mimicking this in a chip. So, but I think this one is, I got a phone call from a grad student in UK. She was trying to build the placenta on the chip because she wanted to look at the immune response, but she was telling me that she was having a lot of problem, but another group actually published last year. They co-culture this endothelial cell with the the placental cell, and then they were able to look at this immune response. So it can be done, and I think also in Europe they're forming this consortium trying to develop this placenta on a chip, trying to look at the communication between maternal side and fetal side. So I thought it pretty interesting. this is becoming a reality. This chip company actually originally they were engineering company. They're full of engineer, right? Building this chip in the lab, but they are changing. The focus right now is not engineering company anymore. They change it to biological company where they service testing for pharma, for FDA. So that's where the money is, because making chip probably will not make them profit. So they basically service it. And then because biologists in the lab, sometimes they probably don't want to spend time building this. So they just pay. When I was in my old company, they actually a pharma like Glaxo and all that contract with us as well, trying to send the drugs, and we build a model for them and we test for them. Yeah, so that's where the money is. So when I came to Thailand, I was trying to think, okay, what can I do? And Thailand is, as you know, grant agency is mostly focused on applications. So we have been trying to develop this organ on a chip, and myself building a human capillary on chip, and Ken actually building a human liver on chip, because Ken expertise is in metabolism. So that's what he's interested in. So why we build this, right? Like I said, the problem for organ on chip is that the reproducibility is very low. And I can see that this is becoming a trend in right now. People try to reduce the animal testing, and 2D is not sufficient anymore. So there are a lot of some lab in Singapore actually work on this. And I just realized China and Japan, Korea has sent a bunch of postdocs to Harvard, and they came home, and then they're actually building this chip facility. So I thought, okay, this is great. So we established this in the lab. So came to Thailand, wrote the grant, hired students and technician, and trained them. It took a while, but we got pretty compelling data. So you can see this is a human capillary on chip that we generate in our lab. And in the panel A, this is just taking the picture from the microscopy. So the picture look pretty flat to us, to you. Basically, the cell we put this is actually three-dimensional structure. The green signal will tell me that the cell are alive, and actually our cell at least we can culture up to day 14, and the structure look pretty stable, and the cell pretty viable. You can't do that with the 2D at all, because after a couple of days they will be full and die. But the advantages of using the chip is that you can culture them for a long time, and the structure is still stable. Yeah. In B, I don't know, you can see here, the screen is pretty bright, but this is a 3D reconstruction of our confocal images. You can see that we got the luminal structure. It's not flat, it's actually have a lumen. And this lumen actually exposed to the flow. So it's mimicking the circulation. Yeah, so. We tested, usually, as you know, our endothelial cell usually form the barrier, right? So macromolecules like albumin cannot get out from the circulation. So we mimicking that function. We put the albumin, not albumin, the dextran that have the similar molecular weight as albumin. And we monitor this for 30 minutes, and we capture picture every 1 minute, and we put them as a video. As you can see here, when we don't have cell, the dye actually leak out into the adjacent channel. But with the 3D capillary that we built, we didn't see anything, or if anything, leaking into the collagen channel here. Yeah. So this telling me that we got pretty tight barrier, mimicking what happened in vivo. Not just tight barrier, it maybe people are gonna ask, like, we have tight barrier, can it be responded to inflammatory cytokine? So we actually pre-treat our capillary with TNF alpha, which is usually making the endothelial cell leak, right? And we found that in red here, we found that the TNF alpha can actually make the green signal leak into the collagen channel, suggesting that our blood vessel can respond to the cytokine as those in vivo. We also can measure cytokine that come out from the chip. And basically, we treat the blood vessel with TNF alpha compared to control vehicle. And we collect the waste media and we assay for IL-6 production. And we found that after TNF alpha treatment, our blood vessel can produce IL-6. So, yeah. Getting to the liver model, this is how our model in 3D, the hepatocyte look like. You can see that it's very different from 2D cell culture, where the 3D cell culture, they from the aggregate. This is 3-dimensional structure, where the 2D is pretty flat. And the cells are alive. And Ken is building the fatty liver model, where he can treat this hepatocyte in a 3D with the fatty acid. And he found a significant increase in fatty acid accumulation in the chip, liver chip, mimicking the fatty liver models. And he used this to test drugs at the moment. Yeah. So we are kind of trying to wrap up this and send out for publications. And I have to tell you that we've been doing this for a while with different personnel in the lab. And the key that I'm happy about is that maybe we do N of 1,000 now, and it's pretty reproducible. So, yeah. So this is what we are happy about. And I think we are ready to take on to servicing this at some point. Yeah. Of course, I have to thank my team and also funding that we have. Currently, next week, actually, I'm defending for PNUC for the second years. This grant particularly is focusing on utilizing the basic science into industrial or local business in Thailand. So hopefully things go well. So, ขอบคุณค่ะ thank you.
ขอบคุณค่ะ Thank you very much for very informative and interesting about the work that you and your team have done, and enlightened me a lot about the basic knowledge that I never ever know that what is working, how it's working. And if any people in the on-site or online want to have question with อาจารย์พิมลรัตน์นะคะ you can raise your hand up or you want to shout it out. It's fine. It's open for discussion ค่ะถามไหมคะอยากถามไหมคะค่ะถามภาษาไทยก็ได้ค่ะอยากถามอะไรถามเลยอาจารย์วิวอยากถามอะไรไหมลุยเลยไม่ no one thing that I want you guys to understand that no question is stupid, okay? Because sometimes the research question come from the stupid question. So if you don't know, maybe you make us, oh, okay, we can research something later on, okay? แพรวฝันหน่วยกล้าตายค่ะเชิญเลยค่ะ
อยากทราบว่า what is IVATA stand for อะค่ะแบบที่บอกว่าเป็น IVATA stand for คำว่าอะไรอะค่ะคือตอนแรกนึกว่ามีตัวย่ออะไรเกี่ยวกับของสูงพยายามดู
No, this is the name for the model. So it try to represent about the endometrium. I think it's about endometrium or
E form endometrium.
Yes, or uterine cavity, something about that. Because they said they are going to make like a menstrual cycle, so it should be endometrium. But this is just yes, we have to look for it later on. But the question related จะต่อไหมคะอยากทำอะไรเพิ่มไหมคะเอาเลย No question is stupid like I said
อยากรู้ว่าตอนนี้มันถึงแบบขั้นที่เราศึกษาแค่ทำสิ่งนี้ให้เหมือน physiology หรือว่ามันเอาไป adapt ใช้กับแบบยาแล้วอะค่ะคือมันถึง คือฟังแล้วมัน
ค่ะตอนนี้
มันถึง
some
ไหนใน
as
worldwide
ค่ะตอนนี้นะคะ US FDA เนี่ยเอาไปใช้แล้ว เอาเข้าไปใช้แล้ว แล้วก็ pharmaceutical company หลายที่พวก big pharma ทั้งหลายอะค่ะเอาไปใช้แล้ว แต่อาจจะไม่ได้ใช้ทุกแบบอาจจะใช้แค่โมเดลที่อย่าง toxicity น่ะใช้แล้วค่ะแล้วก็ยุโรปเนี่ยวันนั้นก็ยังเห็นอยู่นะคะบริษัทบุหรี่ค่ะอาจารย์อะไรนะมอริสที่สวิตเซอร์แลนด์ก็เอาไปใช้น่าจะไปทำรังชิปกันเยอะทีนี้ก็ค่ะอาจจะไม่ได้ใช้ทุกโมเดลแต่ว่าโมเดลที่ well established เช่นรังชิป liver chip เนี่ยมีการใช้แน่ๆแล้วค่ะในฟาร์มาเริ่มแล้วกฎหมายของ US ก็มาแล้วด้วยก็น่าจะใช้เยอะขึ้นไปอีก
ก็มีคำถามอีก
ประเทศเราอะค่ะอาจารย์ทำเป็น capillary ใช่ไหมคะ
ค่ะ
แล้วมันกำลังจะ
ตอนนี้
มันจะถึงไหน
ตอนนี้
แล้วมันมีที่อื่นแบบอื่น university
ตอนนี้ที่เมืองไทยเนี่ยเท่าที่คุยกับหลายคนเนี่ยก็มีเอนจิเนียร์เขาพยายามจะทำชิปที่ประเทศไทยเนาะแต่ว่ามันยังทำไม่ค่อยได้แล้วก็ยากทีนี้พอดีตัวเองมาจากบริษัททำชิปก็บอกเขาว่าอย่าไปทำเลย ก็ไม่เพราะว่ามันรู้สึกเสียเวลาเพราะว่าเท่าที่ตามฟิลเนี้ยคือการทำ engineering perspective มันเริ่มตันแล้วมันดีไซน์ให้ยากกว่านี้ไม่ได้ละเพราะว่าถ้ายากเกินคนใช้ก็ใช้ไม่เป็นอีกก็ไม่รู้จะทำไปทำไมก็เลยบอกว่า biologist เนี่ยแหละต้องเอามาใช้แล้วก็เราต้องเอามาใช้แล้วเพื่อเป็นการ validate ด้วยค่ะอาจารย์ว่า reproducibility เนี่ยมันเอาไปใช้ได้จริงเพราะว่าถ้าอยู่ reproduce ไม่ได้เนี่ยอยู่ก็ไม่มี product นะอยู่ก็เอาไปใช้ไม่ได้มันก็จะกลายเป็น academic ตีพิมพ์ไปเรื่อยๆถูกไหมคะเขาก็จะตอนนี้เทรนด์ก็คือเน้นไปที่การนำไปใช้แล้วก็ทำให้เกิด reproducibility เยอะๆค่ะ
มี question ออนไลน์
ที่เมืองไทยตอนนี้เขาที่เข้าใจตัวเองยังเป็นคนแรกอยู่นะคะแต่อันนี้แหล่งทุนบอกมาก็เลยเราก็ทำต่อไปเนาะแล้วเราก็เดี๋ยวสักพักเขาเปิดตัวเดี๋ยวเราแป๊บนึง
อันนี้มีคำถามจากออนไลน์นะคะใช้ชื่อว่าทัศนีเลิกสุทธิรัตน์นะคะอาจารย์ถามว่า if you are talking about they calling how many chips from one organ should be a good representative to test particular task for example like a toxicity
how many chips ใช่ไหมคะทีนี้ตัวเองเนี่ยอันนี้ถามดีมากนะคะเพราะว่าทุนปีแรกเนี่ยก็คือเรื่อง testing นี่แหละเรื่อง toxicity testing นะคะตามปกติเนี่ยเราจะทำ positive control แล้วก็ calculate พวก C factor ซึ่งเป็นการ validate ว่า assay เราดีหรือไม่ดีนะคะทีนี้ปกติ cut point ที่เขาใช้อันนี้อีเมลมาคุยกันต่อได้นะคะจะอยู่ที่ประมาณ 0.2 แล้วคือว่าปกติเนี่ยเราต้องทำเทียบกับ 2D ด้วยแต่ว่าในปัจจุบันเท่าที่หนูทำอะค่ะก็คือ n of ประมาณ 10 เนี่ยก็น่าจะก็คิดว่าถ้า clear cut มันก็ significant แล้วมันก็ใช้ได้แล้วตอนนี้เรา calculate IC50 ด้วยซ้ำเวลาทำการทดลองค่ะอาจารย์ก็คือทำ different doses แล้วก็ดู IC50 ด้วยนะคะอันนี้คงต้องดูค่ะว่า sensitive แค่ไหนนะคะ assay ที่อ่านอยู่ทุกวันนี้ตัวเองทำเนี่ย positive control ก็ค่อนข้าง sensitive ทีเดียว
ค่ะมีคำถามจากออนไลน์อาจารย์ชื่นกมลค่ะเรียนเชิญค่ะอาจารย์
ขอบคุณอาจารย์ที่มาเปิดโลกนะคะพอดีเคยได้ยินเกี่ยวกับออร์แกนนอยมาบ้างค่ะแต่ว่าทีนี้อยากถามว่าอันนี้อาจารย์ทำเป็นเซลล์ปกติใช่ไหมคะแคนเซอร์เซลล์อาจารย์ได้
ค่ะยังไม่ได้ทำที่เมืองไทยค่ะอาจารย์ตอนที่อยู่ต่างประเทศเนี่ยทำกับเพื่อนเขาพยายามที่จะทำพวก angiogenesis ไปที่ tumor ค่ะอาจารย์ตอนนี้มีคนทำเยอะแล้วก็ที่ต่างประเทศนะคะที่เมืองไทยยังไม่คิดว่ายังไม่ค่อยมีคนทำตัวเองยังทำprimary endothelial cell เป็นเซลล์ปกติค่ะยังไม่ได้มีการ modify อะไรทั้งนั้นยังไม่ได้ทำ genetically modify อะไรทั้งนั้นส่วนliver cell ที่เราใช้ก็ยังเป็นhepatocyte ธรรมดานะคะเป็นเซลล์ไลน์ปกติเพราะเราต้องการ output throughput ที่เยอะๆเพราะว่าพอทำ primary จริงๆถ้าเกิดไปเอาเซลล์จากคนไข้เนี่ยตอนนี้ส่วนใหญ่ในฟิลเขาจะไปทำพวก iPSC กันเยอะนะคะตอนนี้ก็เป็นอีกสเต็ปหนึ่งค่ะอาจารย์ตอนนี้ยังเป็นเซลล์ปกติอยู่ค่ะ
iPSC นี่คืออะไรอะคะ
stem cell ค่ะอาจารย์
อ๋อ
ค่ะค่ะ
ค่ะ อาจารย์ขอบคุณค่ะ
ค่ะ
I just wonder about I just wonder about the price, the cost that we are going to spend for one chip, because when we are tested, for example, like you said, we don't need a lot of population to test about the chip. About 10, it should be all right. But the chip itself, it have a lot
Okay,
of cost
yeah.
as well.
so ค่ะใช่ค่ะแพงกว่า 2D ค่ะอาจารย์อันนี้ต้องบอกว่าแพงกว่า 2D นะคะแต่ว่าไม่ได้แพงเท่า animal ไม่ได้แพงเท่า animal แล้ว animal มันต้องมี facility ใช่ไหมอันนี้ไม่ต้องค่ะต้องทำในห้องแลบธรรมดาค่ะ
And another question is how much, I mean, how long, and we can shorter time when we try to test it on chip first before we going to on the live model, like animal model.
ค่ะ you mean หมายถึงเวลาที่ใช้ใช่ไหมคะคือตอนนี้เขาก็คิดกันไว้ว่าถ้าสมมติว่าผ่านชิปไปแล้วเนี่ยเราก็มั่นใจได้ระดับหนึ่งทีนี้เขาก็มองว่ามันอาจจะเราก็ยังอาจจะต้องใช้ animal model test อยู่แต่ว่าเราอาจจะไม่ต้องใช้จำนวนของสัตว์เยอะเหมือนแต่ก่อนละเพราะว่าชิปเนี่ยมา provide some confidence ว่ามันจะเหมือนกันในคนมากกว่าอันนี้ต้องบอกอาจารย์ว่ายังอยู่ใน ongoing ของที่ต่างประเทศค่ะ
ค่ะขอบคุณค่ะอาจารย์อันต้องบอกว่าอันนี้เป็นเรื่องที่ใหม่มากสำหรับเราซึ่งอย่างที่บอกว่าอาจารย์คือทีมแรกใน University ในเมืองไทยต้องดีใจที่เราได้บุคลากรที่เรากลับมาอยู่ในประเทศเราแล้วก็ตรงนี้คืออาจารย์อันนี้ถือว่าเป็น product ที่เกิดขึ้นของในเมืองไทยเองแล้วเราก็เดี๋ยวต่อไปมันก็จะมีในเรื่องของ patent หรืออะไรก็แล้วแต่
คือตอนนี้ก็มีคนเริ่มเอาเซลล์ชนิดอื่นมาให้เหมือนกันที่เขาอย่างที่เมืองไทยก็จะมีบางกลุ่มที่เขาแบบทำเซลล์ไลน์ไว้ซึ่งอาจจะมี advantage ที่เหนือกว่าเซลล์ที่ขายตาม commercial ทั่วไปก็มีคนมาแบบเอาไปใส่ชิปให้หน่อยสิอยากรู้อะไรเงี้ยเอาไปเผื่อเราจะ patent ได้นู่นนี่นั่นอะไรเงี้ยตอนนี้เราก็อยู่ใน process เรา obtain เซลล์มาแล้วแต่เรายังไม่ได้ทำการทดลองเนื่องจากเราก็ handful ด้วยการทดลองนะคะแต่ว่าอันนี้เป็น next step ต่อไปนะคะก็มีอาจารย์บางท่านตอนนี้ฟิลมันเริ่มแต่ว่ามีคนเอาออร์แกนอยด์ใส่เข้ามาในชิปเหมือนกันซึ่งตอนนี้ตัวเองยังไม่ได้มีเวลาไป explore แต่ว่าที่ต่างประเทศเริ่มมี publication ออกมาแล้วทีนี้ก็เลยออกเมืองไทยเนี่ยมีคนทำออร์แกนอยด์หลายกลุ่มเราก็คิดเหมือนกันว่าเราจะมาใส่นะคะ
ค่ะก็ต้องบอกว่าอีกเรื่องหนึ่งก็คือว่าคือในเรื่องของที่อาจารย์พูดไปเมื่อตอนแรกนะคะ it's been about 14 days you can culture 3D for 14 days at maximum
ของเคนอย่างเงี้ยของ liver chip เนี่ย14 วันแบบนิ่งมากไม่มีปัญหา stable มากเพราะถ้าใน 2D เนี่ยนะคะอาจารย์ liver cell เนี่ย3 วันก็ไปหมดแล้วอะค่ะทีนี้เราก็สามารถที่จะเอามาเทสเรื่อง chronic tox ได้อย่างแบรนด์เราที่ปิดไปแล้วนะคะเราก็เอามาเทสสารที่เป็นพวก natural compounds นะคะที่ทาง chemist เขาไป modify กลุ่มแล้วเราไม่รู้ pharmacological activity เลยเราก็เอามาใส่ในชิปนะคะแล้วก็ดูเรื่องของ toxicity นะคะก็เราสามารถที่จะ assess พวก chronic tox ได้ดีนะคะในขณะที่ 2D เนี่ยเราจะทำเรื่องพวกนี้ไม่ได้นะคะ
จริงๆเรา quite surprised about the EVATA because cycle is about 28 days so you have to make it alive for 28 days difficult but we need to research on it thank you for tell us about this maybe we can explore later on it's time for us now is 9 o'clock and 1:09 so I would like to thank อาจารย์พิมลรัตน์ and อาจารย์ Kenjiro for the talk about their work on about the novel 3D in vitro organ models in Hermatipedi in Jacqueline Narubodin นะคะ and we hope that we can have one more in a specific topic and also about the collaboration in the future. Thank you very much for having invited accept our invitation and then so we will discuss later on. And I would like to close the morning activity today. And could you please give a hand for Ajahn Pimonrat, please? Thank you very much for the audience online and here on site as well. ขอบคุณค่ะ